there! Ready for a wellness adventure? Let's explore the world of health together!
Have you ever wondered what's really going on inside your head? Right now, as you're watching this, a 3-pound universe of tissue is processing light, sound, and a million other things. That's your brain, the most complex object we know of. Today, we're going on a grand tour of this incredible organ, exploring its major regions and uncovering what makes you you. So get ready to meet your own command center. Let's start with the big picture. The brain is divided into 3 main parts: the cerebrum, the cerebellum, and the brainstem. The largest part, the cerebrum, is what you probably picture when you think of a brain, that wrinkled, grayish-white outer layer. This is where the magic of higher-level thinking happens. The cerebrum is split into 2 hemispheres, the left and the right. You've probably heard about being left-brained or right-brained, right? While it's a bit of an oversimplification, the 2 sides do have specialized jobs. The left hemisphere is typically dominant in language, logic, and analytical thought. It helps you understand words, solve math problems, and make logical sequences. The right hemisphere, on the other hand, excels at creativity, spatial awareness, recognizing faces, and interpreting emotions. These 2 halves are constantly talking to each other through a thick bundle of nerves called the corpus callosum, ensuring they work together as a unified team. Now let's zoom in on the cerebrum's surface, the cerebral cortex. This is where all the wrinkles are, and for a good reason. These folds, called gyri and sulci, dramatically increase the surface area, allowing more neurons to be packed in. More neurons mean more processing power. This cortex is divided into 4 main lobes, each with its own set of responsibilities. First up, at the front, is the frontal lobe. This is your brain's CEO. It's in charge of planning, decision-making, problem-solving, and personality. When you re-weighing the pros and cons of a decision or controlling an impulse to eat that extra slice of cake, your frontal lobe is hard at work. It's the last part of the brain to fully mature, which is why teenagers can sometimes be a bit impulsive. A specific area here, called the prefrontal cortex, is the seat of your personality and complex behaviors. Moving back from the frontal lobe, we find the parietal lobe. This lobe is the master of sensory information. It processes touch, temperature, pressure, and pain from all over your body. It's also crucial for spatial awareness, knowing where your body is in space and navigating your environment. When you effortlessly catch a ball or parallel park your car, you can thank your parietal lobe for integrating all that sensory and spatial data. Next, on the sides of your brain, just behind your temples, are the temporal lobes. These are the auditory processing centers. When you listen to music, hear someone's voice, or recognize a familiar sound, your temporal lobes are firing away. But that's not all. They also play a vital role in memory, especially in forming and retrieving long-term memories. Deep inside the temporal lobe is a structure called the hippocampus, which is absolutely essential for creating new memories. Without it, you'd be stuck in the past. Finally, at the very back of your head, we have the occipital lobe. Its job is simple, but incredibly important: vision. Everything you see is processed here. Light enters your eyes, signals are sent down the optic nerve, and the occipital lobe decodes these signals into the vibrant 3-dimensional world you perceive. It identifies shapes, colors, and motion, allowing you to recognize a friend's face from across the room or appreciate a beautiful sunset. It's amazing to think that what you see is actually a construction built in the back of your brain. So we have the 4 lobes of the cerebrum: frontal for thinking, parietal for sensing, temporal for hearing and memory, and occipital for seeing. But we're not done yet. Let's journey deeper beneath the cerebrum. Tucked away under the cerebrum, at the back of the brain, is the cerebellum. Its name means little brain, and it looks like a smaller, more compact version of the cerebrum. While it's only about 10% of the brain's total volume, it contains over half of the brain's neurons. The cerebellum's main gig is coordination, balance, and motor control. When you ride a bike, play a musical instrument, or even just walk without stumbling, your cerebellum is orchestrating all those smooth, precise movements. It learns and automates actions, turning conscious effort into unconscious habit. Now for our final stop on this tour, the brainstem. This is the oldest and most primitive part of the brain. connecting the cerebrum and cerebellum to the spinal cord. Think of it as the brain's life support system. It controls all the essential, automatic functions that keep you alive without you ever having to think about them. Things like your heartbeat, breathing, blood pressure, and even swallowing. The brainstem is always on, even when you're asleep, making sure your body keeps running. It's also the conduit for signals traveling between your brain and the rest of your body. Within this complex system, we also have the limbic system, often called the emotional brain. It's a collection of structures, including the hippocampus we mentioned earlier, and another key player called the amygdala. The amygdala is your emotional watchdog, processing feelings like fear, anger, and pleasure. It's what triggers your fight or flight response when you perceive danger. This system links our emotions to our memories and actions, profoundly influencing our behavior and how we experience the world. So, there you have it, a whirlwind tour of your brain's major regions. From the executive decisions of the frontal lobe to the life-sustaining work of the brainstem, every part works in perfect harmony. It's a network of billions of neurons firing in complex patterns, creating your thoughts, your emotions, your memories, and your very sense of self. The brain is not just a biological machine. It's the source of human consciousness and creativity. The next time you learn a new skill, laugh at a joke. Or simply take a breath. Take a moment to appreciate the incredible orchestra of activity happening inside your skull. It's the most powerful tool you'll ever own. For many years, the brain was often described as a collection of separate centers, each responsible for a single function. Early neuroscience textbooks sometimes label areas as the memory center. Modern research supported by national neuroscience institutions has shown that this description is incomplete. Most mental functions emerge from networks of interacting brain regions rather than isolated locations. A network in the brain refers to a group of regions that communicate with each other regularly while performing related tasks. These regions may be physically distant, but connected through long bundles of nerve fibers. They activate together in coordinated patterns. They produce specific cognitive or behavioral functions. This network organization allows the brain to process information efficiently and adapt to new situations. One reason networks are necessary is that complex behaviors require multiple types of information at the same time. Consider the act of speaking. Producing a sentence involves selecting words, planning motor movements for the mouth, monitoring auditory feedback, and adjusting timing. According to research from public university neuroscience laboratories, these processes involve cooperation between frontal cortex regions, temporal language areas, motor cortex, and auditory processing networks. No single region performs all these tasks independently. The same principle applies to perception. When you see an object, different visual features are processed in separate cortical areas. One group of neurons analyzes shape, another processes color, and another detects motion. These signals are then integrated into a coherent perception. Government-supported visual neuroscience research demonstrates that object recognition occurs through interaction between occipital visual regions and higher cortical areas involved in memory and categorization. Memory is another function that depends on distributed networks. The hippocampus plays a central role in forming new memories, but it does not store them permanently by itself. Instead, it coordinates communication between multiple cortical areas that represent different aspects of an experience. For example, remembering a birthday celebration may involve visual cortex representations of faces and decorations, auditory cortex representations of voices and music, and emotional processing from limbic circuits. The hippocampus helps link these components together into a coherent memory trace. Emotions also emerge from networks rather than single structures. The amygdala contributes to detecting emotional significance, but emotional experience involves interactions with the prefrontal cortex, hypothalamus, brainstem, and sensory regions. Public health research on emotional regulation shows that the frontal cortex plays a major role in interpreting and regulating emotional responses generated by limbic structures. This interaction helps explain how people can adjust emotional reactions based on context and reasoning. Attention provides another example of network organization. When someone focuses on a task, activity increases in frontal and parietal regions that coordinate attentional control. At the same time, sensory areas processing relevant information become more active, while unrelated regions become less active. According to national cognitive neuroscience research programs, these coordinated changes allow the brain to prioritize important information and reduce distraction. Scientists often identify networks using brain imaging techniques that measure patterns of activity across regions. When multiple areas consistently activate together during certain tasks, researchers infer that they form part of a functional network. These networks can be observed even when a person is not performing a specific task. In resting conditions, certain brain regions still show synchronized activity, indicating ongoing communication. One widely studied example is the default mode network. This network includes regions in the medial frontal cortex, posterior cingulate cortex, and parts of the temporal and parietal lobes. According to neuroscience research supported by national health agencies, this network becomes active when individuals engage in internal thought processes such as remembering past experiences, imagining future events, or reflecting on personal information. Another important network is the salience network. This network helps detect important or relevant stimuli in the environment. When something unexpected occurs, such as a sudden loud sound, salience network regions help shift attention toward that event. This allows the brain to prioritize information that may require immediate response. The executive control network, often involving parts of the frontal and parietal cortex, contributes to planning, decision-making. When someone solves a math problem or plans a route through traffic, this network helps maintain goals, evaluate options, and guide behavior. Public cognitive neuroscience research emphasizes that executive control emerges from coordinated activity across these regions rather than a single command center. Network organization also explains why damage to one brain area can produce complex patterns of impairment. If a region serving as a communication hub within a network is disrupted, the flow of information between other areas may be affected. For example, injury to frontal regions can influence decision-making, emotional regulation, and working memory because these regions coordinate communication among several networks. Another advantage of network organization is flexibility. Because functions are distributed across multiple regions, the brain can sometimes compensate when one area is damaged. This capacity for reorganization reflects neuroplasticity, a concept supported by extensive research from national neuroscience institutes. Learning also depends on network changes. When someone practices a skill repeatedly, communication between relevant regions becomes more efficient. Synaptic connections strengthen along frequently used pathways. Over time, tasks that initially required extensive conscious effort may become more automatic as network coordination improves. This process explains how complex skills such as playing a musical instrument or driving a car become easier with practice. Network interactions also help explain how mental states change. During focused work, executive networks may dominate activity patterns while default mode activity decreases. During rest or reflection, the default mode network becomes more active. These shifts illustrate that brain function is dynamic rather than fixed. Networks continuously adjust depending on goals, environment, and internal conditions. It is also important to understand that networks overlap. A single brain region may participate in several networks depending on context. For example, parts of the frontal cortex contribute to emotional regulation, attention control, and decision-making. Their role depends on which other regions they are interacting with at a given moment. Modern neuroscience increasingly focuses on these patterns of connectivity rather than isolated structures. Mapping connections between regions helps researchers understand how complex cognitive functions arise from coordinated activity. According to large-scale brain mapping initiatives funded by public research agencies, studying connectivity provides deeper insight into mental processes than examining single areas alone. Understanding network organization also improves interpretation of brain imaging results. If one region shows increased activity during a task, it does not mean that region alone produced the behavior. Instead, it likely acted as part of a larger coordinated system. Interpreting brain activity in terms of networks prevents oversimplified conclusions about centers for specific abilities. In summary, most brain functions arise from networks of interacting regions rather than isolated centers. Language, perception, memory, emotion, and decision-making all depend on coordinated communication across multiple areas. Networks allow the brain to integrate different types of information, adapt to changing environments, and reorganize after learning or injury. Understanding this network-based organization provides a more accurate picture of how the brain produces complex behavior and mental experience. Modern neuroscience relies on several technologies to study the structure and activity of the human brain. These tools allow scientists and clinicians to observe brain anatomy, measure neural activity, and detect abnormalities. According to national medical research institutions, commonly used brain imaging methods include computed tomography, CT, magnetic resonance imaging, MRI, functional magnetic resonance imaging, fMRI, electroencephalography, and positron emission tomography, PET. Each method reveals different types of information, and each has important limitations. Computed tomography, commonly called a CT scan, is one of the earliest widely used brain imaging methods. CT scanners use multiple X-ray measurements taken from different angles to create cross-sectional images of the brain. A computer reconstructs these images into slices that reveal the structure of brain tissue. CT scans are particularly useful for detecting bleeding, skull fractures, or large structural changes after injury or stroke. Public hospital systems often use CT imaging in emergency situations because the scan can be performed quickly and is widely available. However, CT scans have limited resolution compared to newer imaging technologies. They show general structural features but provide less detail about soft tissue differences within the brain. CORT also does not directly measure neural activity. It reveals structure, not function. Because CT scans involve exposure to X-rays, medical guidelines recommend using them only when clinically necessary. Magnetic resonance imaging, or MRI, produces more detailed images of brain structure without using ionizing radiation. MRI scanners use strong magnetic fields and radio waves to measure how hydrogen atoms in the body respond to magnetic signals. Because brain tissue contains large amounts of water, these signals can be used to construct highly detailed images of brain anatomy. According to government-funded biomedical research, MRI can detect subtle structural changes in brain tissue, tumors, inflammation, and degenerative conditions. MRI provides a clearer view of brain structure than CT scanning. It can reveal the shape and size of brain regions, differences between gray matter and white matter, and the presence of lesions or abnormalities. Physicians often use MRI when detailed structural evaluation is needed. However, MRI still primarily shows anatomy rather than moment-to-moment neural activity. Functional magnetic resonance imaging, known as fMRI, builds on MRI technology to estimate brain activity. Instead of directly measuring electrical signals from neurons, fMRI detects changes in blood oxygen levels. When neurons in a particular region become active, they require more oxygen and energy. Blood flow to that area increases, and the scanner detects this change. This signal is known as the blood oxygen level dependent signal. Because fMRI measures blood flow rather than electrical activity, it provides an indirect estimate of neural activity. The signal changes slowly compared to actual neural firing. Neural signals occur in milliseconds, while fMRI signals unfold over several seconds. Public neuroscience research emphasizes that fMRI identifies regions associated with tasks, but does not show the precise timing of neural communication. Despite this limitation, fMRI has been extremely useful for studying large-scale brain networks. Researchers use it to observe which brain regions become active during tasks such as reading, remembering, or making decisions. It has helped scientists map functional networks and understand how distributed brain regions cooperate during cognition. Electroencephalography, or EEG, takes a different approach. Instead of measuring blood flow, EEG records electrical activity produced by groups of neurons. Small sensors placed on the scalp detect voltage changes caused by synchronized neural firing. According to national neurological research agencies, EEG provides excellent temporal resolution, meaning it can measure changes in brain activity within milliseconds. This high-time precision makes EEG useful for studying processes such as attention, sleep cycles, and sensory responses. For example, researchers can measure how quickly the brain responds to visual or auditory stimuli. EEG is also widely used in medical settings to monitor seizure activity. However, EEG has lower spatial resolution than imaging techniques like MRI. Because the sensors sit on the scalp, it is difficult to determine the exact location of deeper brain activity. Positron emission tomography, or PET scanning, provides another method for examining brain function. PET involves injecting a small amount of radioactive tracer into the bloodstream. The tracer emits signals that can be detected by the scanner. By tracking where the tracer accumulates, scientists can observe metabolic activity or specific chemical processes in the brain. For example, PET can measure glucose metabolism, showing which brain regions consume more energy during certain tasks. It can also track specific neurotransmitter systems using specialized tracers. Public medical research institutions use PET imaging to study conditions such as Alzheimer's disease, where abnormal protein accumulation can be detected with certain tracers. Because PET involves radioactive materials, its use is generally limited to specific clinical or research applications. It is not typically used for routine brain imaging. PET also has lower temporal resolution than EEG and fMRI, meaning it cannot capture rapid neural changes. Each imaging method therefore provides different kinds of information. CT and MRI show structural anatomy. fMRI estimates functional activity through blood flow changes. EEG measures electrical timing of neural activity. PET measures metabolic or chemical processes. Neuroscientists often combine multiple techniques to obtain a more complete picture of brain function. It is equally important to understand what these methods cannot show. Brain imaging does not allow scientists to read thoughts directly. While certain patterns of activity may correlate with specific tasks, interpreting those patterns requires careful experimental design and statistical analysis. According to public neuroscience guidelines, images showing activated brain regions represent averages across many measurements rather than single moments of thought. Another limitation is that imaging results show associations rather than direct causation. If a brain region becomes active during a task, this does not necessarily mean that region alone produces the behavior. It may be part of a broader network involved in processing the information. This principle reinforces the network-based understanding of brain function discussed earlier. Brain imaging data also require interpretation within the context of experimental design. For example, if researchers compare brain activity while people read words versus viewing blank screens, the difference in activity may reflect multiple processes including vision, attention, and language. Careful control conditions are needed to isolate specific cognitive processes. Medical imaging must also account for individual variation. Brain anatomy and functional patterns differ somewhat from person to person. Public research institutions therefore rely on large sample sizes and statistical models to identify reliable patterns across individuals. These methods help distinguish meaningful signals from random variation. Another important consideration is that brain imaging does not capture the full complexity of neural communication. Many processes occur at microscopic scales, such as synaptic transmission and molecular signaling. Imaging techniques observe larger patterns rather than individual synapses. This means that cellular-level mechanisms must be studied using other methods such as electrophysiology or molecular biology. In clinical medicine, brain imaging is often used alongside behavioral assessment and medical history. A scan can reveal structural changes or abnormal activity patterns, but it does not replace careful evaluation of symptoms and functioning. According to national medical guidelines, diagnosis and treatment decisions integrate imaging findings with clinical observation. In summary, brain imaging technologies provide powerful tools for observing the structure and activity of the brain. CT and MRI reveal anatomy. fMRI estimates functional activity through blood flow changes. EEG records electrical timing of neural signals, and PET measures metabolic or chemical processes. Each technique contributes different information, and none provides a complete picture alone. Understanding both the capabilities and the limitations of these tools helps clarify how scientists study the brain without assuming that imaging alone can fully explain thoughts, emotions, or behavior. Thanks for watching World Wellness Mind, Brain, and Behavior.
Thanks for watching! Hope you liked it!